Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And in what we were discussing about the electrophoresis, and in this particular module we have discussed about the basics of electrophoresis, followed by the vertical gel electrophoresis, horizontal gel electrophoresis, and then in the previous lectures we have also discussed about the different variants of the gel electrophoresis, and then ultimately we have also discussed about some of the research problems where you can be able to utilize the electrophoresis as a tool to answer those questions. And in this particular series, now today we are also going to discuss few more experiments and few more research problems, and while discussing these research problems, you will be able to understand more and more potential of the electrophoresis to solve the research problems related to your work. So, the first problem today what we are going to see is, and this is an old problem what we have discussed before also, that the scientists have discovered a transcription factor and in the skin cells bind to the set of gene X, which is responsible for changing its color due to the exposure to the sunlight. Now, they would like to identify the transcription factor from the skin cells. What they want to do is, if you remember in the previous lecture, what we have discussed, that they were interested to identify the transcription factor, and then on the top they are also interested how the DNA as well as this transcription factor is interacting with each other. So, as we I think discussed that the gene structure is made up of promoter, and then you have the coding regions. So, this coding region is not responsible for binding of the transcription factor, only the promoter region is responsible for binding of the transcription factor. And in this event what will happen is that the DNA is making a complex with the protein, and as a result it is forming a DNA protein complex. Now, if you see how how this interaction can be mapped with the help of the electrophoresis. So, in the experimental design what we have the two molecules, one is DNA, the other one is the protein, and what they are doing is they are interacting with each other to form the DNA protein complex. Now, let us assume that the DNA is of the molecular weight of 1 unit and the protein is also of a molecular weight of the 1 unit, which means the DNA protein complex is going to have the molecular weight of 2 units. Now, let us assume that the DNA, if you do the electrophoresis of these 3 molecules, then the electrophoretic mobility of DNA is X, the electrophoretic mobility of Y is the protein is Y, then and the DNA protein complex is Z. Then the question comes whether the X is going to be bigger to the Z or X is going to be the smaller to the Z, which means whether the DNA is going to be run faster than the DNA what is present in the complex, or whether the DNA what is present is going to be run slower than the DNA what is present in the complex. As obvious that the molecular weight of the DNA protein complex is bigger, so it is actually going to run on a slower side, which means that the DNA is going to have the higher electrophoretic mobility compared to the DNA what is present in the protein. Because as you remember that the electrophoretic mobility in the gel electrophoresis is directly proportional to the charge and the inversely proportional to the molecular weight. So, this means if you have a smaller molecular weight, you are going to run faster and you are going to have a larger distance, you will cover the larger distance, whereas the if you have a bigger molecular weight, then you are going to run the smaller distances. So, this interaction of the protein and DNA is being mapped with a technique or with the assay which is called as the gel mobility shift assay. In this technique, and this technique is relies on the fact that a small DNA has a much higher mobility in gel electrophoresis than what is bound to the protein. So, what you can see is if I am running the electrophoresis or if I am doing the electrophoresis of the DNA alone, the DNA will run up to this point, but if I keep adding the protein molecules, the protein molecules are actually binding to the protein to the DNA, and as a result it is actually reducing its electrophoretic mobility. Now, imagine that the DNA what you are putting it for the electrophoresis is 100 kilodalton, okay, and the protein what we are putting is at 25 kilodalton. Now, see, when you are running the DNA alone, it is actually having a molecular weight of 100 kilodalton, but when you are adding the small amount of protein, it is actually the one molecule of the protein. If the one molecule of protein is interacting with the DNA, the resultant molecular weight is going to be 125 kilodalton. Similarly, if the two molecules of the protein will interact, then it is going to have the molecular weight of 150 kilodalton, and if the three molecules of the protein are going to interact, it is actually going to have the 170 kilodalton as a molecular weight. This means as the more and more protein molecules will bind to this DNA molecule, the resultant molecular weight of the complex is going to be keep increasing, which means you can be able to map the interaction between the two molecules simply by looking at the molecular mass. So, for performing this experiment, what you are supposed to do? You have to first prepare the cell lysate, you have to do the incubation reactions where you are actually going to incubate the DNA plus the protein, okay, and then you are going to done the polyacrylamide gel electrophoresis, and that actually is going to give you the resultant retardation of the DNA bands. The material, reagents, the material and the equipment what you required. So, the first reagent what you required is the labeling reaction mixtures, so that you will be able to label the DNA, because how you are going to monitor the migration of or retardation of the DNA molecule is that you actually going to label the DNA with a radioactive material. So, in this case we are using the P32 labeled oligonucleotides. So, what you are going to do is you are going to take the oligonucleotides, you are going to done the 10x PNK reaction buffers, and then you are going to add the enzyme that T4 polynucleotide kinase, and then you are going to add the alpha 32 labels ATP and the double distilled water, and this labeling reactions is going to be catalyzed, and then it is actually going to label the DNA molecule, which is the oligonucleotide which you are going to use in the gel shift assays. You also need the binding assay buffer. So, the composition of the binding assay buffer is given here, which actually mostly contains the buffers, then you have the reducing agents, and then you have the EDTA, so that you should not you should take care of the contaminating metals and all that. And then you need the resolving buffer which you are going to use for running the page. So, 5x TBA buffer, where you are going to have the tris, boric acid and EDTA, and then the funnel volume what you are going to make up to the 1 liter. And then you need to prepare the recipe for the non-denaturating page gel. So, where you are going to take the acrylamide, water, TB, then APS and timid are going to be added as the polymerization agent, and then you require the sense as well as the antisense oligonucleotides, so that you can be able to design the oligonucleotides. And then in the instruments what you require, you need a gel electrophoresis system and as well as the microfuge. Microfuge means you require a small centrifuge, so that you will be able to pellet down the reaction mixtures. This can be done in a multi steps. So, in the step 1 you are going to prepare the labeled probe. So, the labeled probe can be prepared by multiple ways, and that all we are going to discuss in detail when we are going to discuss the Southern blotting. And so, for time being you can just simply understand that you are going to add the you are going to take the oligonucleotides, you are going to add the radioactivity or radio labeled ATP, and then you are just simply going to add the P and K, and that actually is going to catalyze the reactions to add the radioactive ATP to the oligonucleotides. So, here you are going to take the two oligonucleotide, one is the sense oligonucleotide, the other one is the antisense oligonucleotide. So, that you can be able to design a double stranded DNA. So, for example, we have given you a sequence of the sense and antisense oligonucleotides. So, this is the sense strand, this is the antisense strand which you can use, and this is the sequence which is actually going to bind or which is going to facilitate the binding of the protein. So, that is the region which you have to keep in the center of this particular sequence, so that there will be adequate amount of nucleotides will be available on both the sides, so that the protein could be able to sit on top of this particular sequence and will be able to bind very nicely. Then you have to do the several steps of doing the labeling reactions, and then ultimately you have to purify the labeled nucleotide by the gel filtration column using the G50 columns, and this column is pre-equilibrated with the buffers, so that you will be able to remove the excess radioactivity from the radio labeled nucleotides. So, once your labeled probe is ready, then you have to go to the step 2. So, in the step 2 you have to set up the reactions. So, for the setting of the binding reactions, what you have to do is you have to take up the nuclear protein extracts are incubated with the labeled probe. While setting up the binding reactions, the following components are added in the given order under the ice cold conditions. So, this is very important that you do all these experiments under the low temperature conditions, so that there will be no degradation of the DNA or there will be no degradation of the protein as well, because either of these degradations are going to compromise the overall effects. So, what you need is you need the salmon sperm DNA, which you are going to prepare in a DEPC treated water. So, DEPC treated water is a water which is free of the RNAs, so that there will be no degradation of the RNA if at all the RNA is present, because that actually is going to mask the signal or what you are going to get from the DNA. Then you also need the binding buffers. So, the composition of the binding buffer is already been given in the previous slide. Then you have to prepare the nuclear extracts. So, you have to add the X microliter of nuclear extract. So, that depends on the what kind of activity is present in your sample and all that. Then you require the labeled probe. So, this nuclear extract is actually going to be the source of the protein, because the nuclear extract is going to have the transcription factor. So, that you are interested to test, and then you have to make up the volume with the double distilled water. So, after the addition of all the components over the ice, the tubes are spinned briefly in microfuge, so that you can be able to collect all the reaction mixtures, and then they are incubated for 45 minutes at 25 degrees Celsius. So, that there will be interaction of the transcription factors what is present in the nuclear extract with the oligonucleotide which is we are adding, and then it is actually going to form the complex. In this there will be a variation that for the super shift experiments. So, super shift experiments are the experiment where you are also being interested that which protein is actually binding. So, in those cases what you can do is you can simply add the antibodies directed against a particular transcription factor, so that you will be able to not know only that the proteins are binding, but also which protein is binding. So, those kind of experiments are called super shift experiment, because in a normal gel shift assay what you are going to do is you are going to see a gradual retardation of the DNA or gradual moving of the DNA on the upward side. But in the super shift experiment, as soon as you add the antibodies are going to increase the molecular weight of the complex very high. So, because of that it is actually going to give you additional shift into the retardations. So, the 2 microgram of the respective antibody is added to the binding reactions prior to the addition of the labeled probe. Because of that, it is actually going to label, or it is going to bind the protein or the transcription factor of your interest. So, because of that, it is actually going to show you the super shift, which means in a normal thing what you have is you have a DNA here and then it will gradually go actually. Whereas, in the super shift what will happen is you have a DNA here and then it will show a very large jump in terms of shift, because here you only have the protein, whereas here you have the protein as well as the antibody. So, and that actually is going to increase the molecular weight in a very large quantity. So, it will actually going to add up a lot in terms of the final protein DNA complex what is going to be formed. So, this mixture was incubated for 20 minutes and then the labeled probe is added and incubation are carried out for another 45 minutes. So, the only difference between a normal gel shift assay as well as the super shift assay is that you have to add the antibodies prior to adding the oligonucleotide, so that the antibodies will go and bind to their respective target proteins, and then if those proteins are go and bind to your DNA, they are actually going to show you the super shifts. Then the step 3, you have to do the analysis of the reaction. So, once the analysis incubation periods are over, then what you have to do is you have to take these samples and then you have to analyze them onto the 5% non-denaturating polyacrylamide gels. In each well you can resolve the 20 microliter of the reaction mixture, and to monitor the progress of the gel, a tube containing the labeled probe alone together with the dye bromophenol blue is added in one of the lanes, so that you will be able to see how long the DNA will migrate if the protein component is not available, because that will be your reference point, how much shift of the DNA band you are getting when you are adding the nuclear extracts. Then the 5x TB is used as a running buffer. The gel is run at the 50 volts constant, and it has to be done in a cold condition, so that there will be no degradation of the DNA, because if there will be a degradation of the DNA, that also is going to give you a gradual decrease in molecular weight, and so that also is going to give you a smear like condition. So, if there will be a DNA degradation, what will happen is the DNA will be here and then you are going to see a lot of bands, and that actually is going to mask the actual DNA samples. Even if you have a sample of DNA here, it is actually going to be masked by that degradation of the DNA probe what you have added in the reaction mixture. So, because of that, it is important that you should keep the DNA in a very stable conditions, and you should even when you are resolving the samples, you also should resolve it in a cold condition, so that there will be no degradations. After completion of the electrophoresis, the gel is dried up by wrapping it in the gel wrap, and it is exposed to a sensitive X-ray film for 1 to 3 days. Band can be seen after developing the film and densitometrically analyzed using ImageJ softwares. So, the image analysis anyway we have discussed in a detail, so that you can be able, once you have the digital image, you can be able to utilize many softwares what we have discussed before also, that either you can use the ImageJ or any other commercial softwares what are available from the different companies. Let us see what result you are going to see. So, in the results section, the positive autoradiograph shows that the positive binding experiments, which means what you can see in a typical experiment, that when you are running the probe alone, it is actually going to run in a very far away from your lane. But as you increase or as you put the nuclear extract, what will happen is that band is going to be keep moving towards the upper side, which means the proteins are now binding in a very low quantity, and then ultimately the whole protein is bound, and what you see is actually there will be a separation, which means what you see is the kind of the curve, which means at this stage the whole DNA what you have added is being saturated by the proteins which are present in the nuclear extract. Now, how you can be able to verify this? You can be able to verify this simply by adding the cold oligonucleotides. So, what will happen if you add the cold nucleotides? The cold nucleotide is going to compete with the protein what is bound here, and because of that it is actually going to release the bound oligonucleotide, which is the labeled oligonucleotide. So, what will happen is that you will be keep observing the band at the bottom. So, if you are going to form the protein complexes, and then if you add the cold oligonucleotide, what is mean by the cold oligonucleotide is the unlabeled oligonucleotide. So, that unlabeled oligonucleotide is going to compete with the complex, and it is going to release the radio-labeled oligonucleotide, and in that process you are going to again going to see the reappearance of the radio-labeled oligonucleotide, because that the protein is making a complex with DNA is giving you the protein DNA complex. But if you add the probe which is not radio labeled, so this probe is actually radio labeled, that is why you have a radio labeled complex. So, if you add the probe or the DNA which is not radio labeled, so what will happen is that this protein is going to be fractionate between the labeled probe versus the unlabeled probe. So, ultimately what will happen is it is actually going to start forming the complex with the unlabeled probe, and the labeled probe is now going to be released from the reactions, and this labeled probe is again going to be electrophorate, or it is going to have the similar kind of electrophoretic mobility what the free probe is having. So, that is why you are actually going to see the reappearance of the labeled probe. So, that is a kind of a verification that this is a specific interactions. On the top, if you are more interested, what you can do also is you can simply block the sites which is present onto the DNA as well with the help of different types of antibodies, and in those cases the protein what you are going to add will not be able to interact. So, these are the different way in which you are going to get the result and in which you can be able to verify the results. So, with this I would like to conclude our lecture here. In our subsequent lecture we are going to discuss some more problems related to electrophoresis.